Let me call up the next panel session, chaired by Janice Gabrilove, and the panelists can come forward. And while she's coming up with her team, I'll just say that Janice has been a tremendous resource and support in a lot of activities around clinical trials. Her formal title is Director of Clinical Research Education Programs here, but her activities and her expertise extends far beyond that. It's only natural that once we identify drugs, we think about how to test them in patients and how to get them to needy patients as quickly as possible. That's a theme that Janice and her team will address now. Janice?
Thank you, Scott. If my colleagues could come up to the podium, or to the table, please. So we're very fortunate today as we move from the discovery arena into the clinical development, realizing that this is truly an iterative process and there's constant crosstalk between the clinical arena and the basic science arena in the development of therapeutics, overall development of therapeutics. We're very fortunate today to have 4 outstanding panelists, and I'll be asking each one of them to tell you a little bit about themselves. But I'll just introduce them. We have Daniel Burkhoff, who's Director of Heart Failure, Hemodynamics, and the MCS Research, Cardiovascular Research Foundation. Dr. Gwen Nichols, Site Head, Translational Medicine Oncology at Roche Pharmaceuticals, Research and Early Development, located down the block or downtown in Manhattan here at the Alexander Center. Dr. Nicholas Schork, Professor and Director of Human Biology at the J. Craig Venter Institute, and Gilles Tamagnan, Laboratory Research and Development Director at the Institute of Neurodegenerative Disorders. So with that, I'll ask each of them to tell you a little bit about themselves, and we'll start off thereafter with a general question for all of them. Please.
So good morning. Thank you very much for the invitation to participate in such a wonderful symposium. My name's Dan Burkhoff, and I'm at the Cardiovascular Research Foundation. I started my professional life as a heart failure clinician at Columbia University, where I also ran a translational lab, and obviously our lab focused on development of large animal models of heart failure, and through that experience, we got very involved with companies that were developing different devices for heart failure, in addition to the basic research that we were doing to look at fundamental mechanisms of disease. Through that experience, I got very involved with various industrial partners and became very intrigued by that whole process of developing devices from concept to clinical practice. Got very involved with preclinical work that was required for FDA approvals to initiate studies, and then ultimately assisting companies to run clinical trials in the device world, largely at that time, first-in-man studies that were primarily being done in Europe. And I became so intrigued with that I really decided to switch over to the industrial side, and I spent about 10 years or so working in industry primarily. And we also had the opportunity, at least I had the opportunity to work with several venture firms to also understand how these products are evaluated and funded, what's the decision-making by venture capitalists, by large companies, and became medical director of several of the companies that we started. And with the purchase of the last company that I worked with, I came back towards academia, working now at the Cardiovascular Research Foundation, where I'm really focusing on, still working with a lot of companies on innovative trial designs for heart failure and other cardiovascular device-based therapies.
Gwen?
Good morning. So I began my training at Sloan Kettering, where I had the opportunity to work with your wonderful Dr. Gabrilove. After being on the leukemia service there for a number of years, I was recruited to Columbia as a hematologist, oncologist, and led the heme malignancies program there for a number of years. In that capacity, I also had a laboratory where I did translational work that would allow me to do investigator-initiated studies and investigator-sponsored studies. And so had some early work in biomarkers to understand how that might allow academicians to get into the pharma world. At some point, I got tempted and moved to the pharma world, and I now work at Roche in their early development division, where my role is dual. It is actually a role where I supervise a number of molecules that are about to enter into humans, and then I also work with preclinical colleagues to try and figure out, once you have a clinical candidate selected, what do you need to be able to prove that that has some benefit in humans? And I think increasingly, we're not looking for changes in CT scans anymore. With our molecules becoming increasingly specific, we really need to have proof of mechanism studies and understand the target and the biology well enough that we can say, yes, this is actually doing what we think it's doing, prior to expecting a single monotherapy to have a dramatic effect in phase 1 oncology and hematology patients. In this capacity, I also get to work with a lot of academicians. We have a very exciting study with one of my molecules here with Ron Hoffman and John Mascarenhas in myeloproliferative disorders. And I also get to work with both academics and small companies trying to look at whether or not we should partner these assets to move them forward within Roche.
Great. Dr. Schork, please.
Great, so it's a real pleasure to be here. I'm Nicholas Schork. I have appointments at JCVI, as was mentioned, but also UCSD and TGen in Phoenix, Arizona. Prior to my those positions, I was a co-director at the Scripps Research Institute, organizing a translational program. Previous panel had talked about academic drug discovery, so I lived that for 7 years at TSRI. Two themes that I've sort of been championing, both in my publications and speaking engagements, having to do with clinical trials, are that I believe clinical trials should have as their focus getting patients better, not necessarily vetting compounds. I think there's a not-so-subtle distinction. Secondly, I think that the future of treating patients is going to be rooted in algorithms that match treatments to patient profiles. So what will actually be vetted are the algorithms themselves, again, not necessarily the compounds.
So good morning. Thank you for inviting me here. So I'm Gilles Tamagnan. In addition of my role at IND, so Institute for Neurodegenerative Disorders, I'm overseeing all the research and all the evaluation of novel PET tracers. So we are in a company called Molecular Neuroimaging, MNI, and we are developing PET and SPECT imaging tools that can be used in clinical studies, either for in neurology, psychiatry, but also now in immunology. So we have a setup where we have chemistry team, biology, animal facility, where we can develop those PET imaging agent or those imaging agent with the goal always to move as quickly as possible to evaluate them in a clinical setting. So we have our own clinical group, and we also work with every country pretty much in the world, where we can set up imaging centers, and we do set up imaging centers around the world where a pharmaceutical company like Roche or Takeda or whomever else wants to run a large phase 3 study where imaging is an inclusion criteria for that study to make sure that only the patients that are going to be benefiting from the drug can be enrolled in the study. And that has two aspects. One is ethical, to make sure that we treat only the people who are going to be benefiting from the drug, but also from a pharmaceutical point of view, it's removing the noise that people without the disease will have in overall clinical trial. So if we are talking about Alzheimer's disease, roughly 25%, 25 to 30% of the people recruited by the best neurologist will not have amyloid deposit in the brain. That includes noise in the study.
Great, thank you so much. So I think some of you have touched on this, but perhaps can expand a general question. What do you see as some of the important drivers that are really mandating the need for newer or innovative trial approaches? And what are some of the resources and expertise that both pharma and academia need to think about in terms of implementing the appropriate infrastructure for fostering those largely first-in-man type of approaches? Would you like to start?
That's a broad, very broad topic. I think, well, in the cardiovascular device world, which I can only speak to, the mortalities have dropped quite a lot for most cardiovascular diseases, thanks to, for example, very early coronary interventions. But that is leading to a much larger population of heart failure, for example, patients with heart failure. And these patients are very plentiful, but the therapies, the medical therapies we have are also improving. So life expectancy is improving. This makes it very difficult to do clinical trials where mortality, which is kind of the gold standard, is the clinical trial endpoint. So what we struggle with is defining appropriate surrogate endpoints that can be used to form the basis for designing clinical trials that are appropriately powered but are not thousands and thousands of patients. So I think having appropriate surrogate endpoints for the big trials, for the pivotal trials, is a real urgent need in heart failure, for example, and specifically.
Great. Gwen, do you want to pick up on that? You started to talk a little bit about that.
Yeah, so I think there are a couple of obvious things. The length of time that it takes to get a drug through development and the cost, which leads obviously to the cost of drugs for patients, is no longer sustainable. And we've talked about that quite a bit in the last day and a half. I think the endpoints are important, but the trial designs are important. And some of this comes from understanding, and maybe this is how we partner best with academia, the depth of the biology. Because if we start with compounds because they have an interesting target, we're fooling ourselves if we think that these diseases are only one target and that they fit all patients. And I think this is precisely what Nichols was saying. And so we need to understand assays and clever ways to answer the question, is this doing what we think it should be doing, with the fewest number of patients, or maybe no patients, before we enter into human beings, and to have the appropriate assays and the appropriate biomarkers to look at to answer the question as quickly as possible in a novel way, so that we don't bring all these phase 2 failures forward.
I would agree with that. I think from my perspective, we could maybe talk about different designs. One design that's getting a lot of attention are the basket and kind of bucket trials, right? Where you start out with some assumption about how you can take, say, a tumor profile, and on that basis, decide what treatment bucket to put patients. That assumes that your algorithm for matching the drugs to the patients is rooted in something. If that biology is off, then the algorithm won't work. So clearly, designs that try to match, say, tumor profiles to treatments rest on certain assumptions that may or may not be valid. So what is ultimately tested are not the individual drugs, but the assumptions upon which the matching is based. And this has implications for the trial design, because you could imagine if during the trial you learn that one or another drug isn't doing well, you may minimize the number of people that are, say, randomized to that basket. But what that means is that the people treated at the end of the trial might actually get benefit from a better algorithm in the early stages of the trial. This could impact how one interprets the data. You might need to weight patients at the end of the trial more heavily than at the beginning of the trial. So there's a number of kind of even statistical issues that kind of plague even these designs that I think the community is going to have to tackle as we go on.
So for me, I think one of the, besides what everything has been said already, in CNS especially, I know that most of the company, and large and small, now do not go into even a phase 1 study until they have a way to show target engagement. That is something that is critical, and there are many ways to do it. Clearly, we champion one way, but that is not the only way to go for it, because sometimes it's not possible because of a lack of target or density of target for technical aspect here. But I think it's, and again, I go back to the ethical issue that I think it's not a good thing to me to engage a phase 1, phase 2, or even further. If you remove the money issue of the cost of enrolling people, if you cannot show that the target the compound you have developed, either small or large, is not going to go, is not going to have any effect because it doesn't go where it's supposed to go. And I think now, today, in 2016, we don't do that much as a community, but I can tell you 10 years ago that was very common, where drugs were going into phase 3 without any knowing if the compound was going to the brain. And there are many, I was talking with Robert earlier, many companies can give a lot of stories about that.
So let me follow up on 2 points that have been raised. The first notion is validating target interaction in the clinical setting in first-in-man approaches, and the FDA has developed the EIND, or exploratory trial, phase 0, and yet the adoption or the utilization of phase 0 study designs has not really taken off. Do you care to comment on why that has or has not occurred?
Yeah, maybe.
Would you like to start? Yeah.
Yes.
Okay,
so we have a lot of experience in IND submissions. So we have something like close to 70 INDs between mixed full INDs or exploratory INDs. So I think over the past 10 years since FDA has presented this approach, and I'm going to talk only about imaging here because that is really for me the poster child of what is an exploratory IND, because we're talking about microdosing, and imaging by definition is microdosing. And I think we, over the past 10 years, we have evolved on the way of using exploratory INDs. And I think the main, for me, the main purpose of it is that you can, in a single application, you can test many compounds which are very similar in structure. And then the question is, what is similar in structure with only one tox package? And you can do that with the same subjects or different subjects. It's a way of being efficient in identifying the best compound for the question you want to ask. To me, if you do not have 2, 3, up to 4 compounds to test, I will recommend that you go directly in a full IND, because the amount of tox you will acquire is very is similar. So you don't reduce the time, you don't reduce the cost, but it's I think it's a question of fine-tuning what you want to get.
Great. Gwen, did you want to comment at all on that?
Well, I think probably for oncology, the main problem has been that a lot of the target studies we might do in a phase 0 setting are not as relevant for the doses and the schedules and the type of toxicity that we anticipate we might see in the first-in-man studies. And so I think it's been, unfortunately, not adopted as often as it should be.
Nicholas, you raised a question about some of the travails of basket trials, and I'd like to ask about some other aspect of these. So basket trials, for those less familiar, have been recently launched and are based on identifying specific molecular phenotypes and are largely disease or organ-specific agnostic. And yet clinical investigator expertise, patient stakeholders, and often pharma, along with CRO alignments that identify participating sites, are in fact are disease-focused. So what are some of the things we need to do to foster these types of designs if we feel they will be informative? And how do we promote realignment or incentives for investigators that are in fact disease or organ identified to be more cross boundaries?
That's a great question. I think, one of the fundamental assumptions about these basket or bucket trials is that you're going to get the biology right. So when you profile the tumor, you're going to be able to identify the overexpressed targets, whatever it is. So one concern that kind of plagues these trials is kind of the garbage in, garbage out principle. Are you in fact measuring the right things? And to what degree do you have to get that right in order to put together an appropriate basket or bucket trial? So for example, I was involved and still am involved in the Stand Up to Cancer Melanoma Dream Team trial, where we're actually trying to show that genomic profiling can result in better patient outcomes so that one could identify more appropriate drugs. We probably spent 2 years just proving to the FDA, because we engaged the FDA in the conduct of the trial, showing them that we could actually reliably assess the tumor profiles and do it consistently. So I think before one even wants to consider jumping from one tumor type to another, you have to make sure that you've got the right targets at least specified. So if it's overexpression of EGFR, whether that's in the context of colorectal cancer or some other tumor, you need to get it right. You need to pay attention to the other sorts of players that might mitigate against the success of certain drugs. All that needs to be taken into consideration. I think right now, many of the basket and bucket trials, I don't want to critique any one in particular, don't pay enough attention to these sorts of issues. And so you have things like the Shiva trial, which for all intents and purposes was a failure, didn't really show any improvements in the people who had genomically profiled and guided drugs. Maybe there was some flaw in the way the matching algorithm from the drugs to the tumor profiles was leveraged, or maybe they didn't take into consideration all the mitigating factors that might plague the prescription of one drug or another. So, there are a number of different issues that I think we need to overcome.
So all these points are very valid, but assuming, to play the devil's advocate, that a basket trial in some arenas is actually valid, are there any models or any thoughts about how to enable us to move from a very stylo, disease organ specific focus of expertise to a more molecular genomic expertise in terms of the investigator pool?
Sure. Well, you certainly need the infrastructure to carry out these studies, and I'm not sure if this is where you're going, so if not, just pull the plug and ask me to revise things. But there are many groups out there that are trying to build appropriate infrastructure for conducting these trials. I've had the pleasure of working with an organization called the MedC organization. I don't know if you're familiar with them. You can look them up online. But this is an organization that's trying to build pre-competitive infrastructure, engage all pharma to really put together the resources to conduct the sorts of trials that we're discussing. The basic intuition is for all the pharma out there that are developing drugs to maybe treat lung cancer, they're going to have to engage a CRO, come up with the appropriate case report forms, partner with academic medical centers. That costs millions, if not billions of dollars. So if there was infrastructure to recruit patients, do the tumor profiling, and then steer them to the right, small single-arm trial or whatever it is, then maybe there would be enormous savings. Of course, it would also require that the profiling be done reliably. So again, I think part of this is infrastructure, and there are efforts underway. Whether MedC is ever going to get off the ground is an open question, but it's one attempt to overcome the shortcomings that kind of plague the use of these sorts of trials in more sophisticated ways.
Great. I'd like to switch gears a little bit, and maybe Daniel, you can start off with adaptive designs, which have been touted as an important newer approach in the development of devices and the development of therapeutics to more rapidly assess efficacy, and yet these approaches have only gradually been implemented. Do you care to comment on why there's been a limited, widespread adoption of this and what's really needed to allow these types of novel approaches that are innovative and make intuitive sense based on probabilities that to go forward full speed ahead?
Well, I think in the device world, the sponsors like to design trials which are very limited, and they know the endpoints, they know the number of patients, and they know the duration of the study. So in a sense, when you have adaptive types of designs, there are certain unknowns that you can't really account for, especially if you're a startup company where you have a budget, you have a timeline, and if you exceed those limits, then you're in trouble, big trouble. So a lot of studies, sponsors really need to have a much more solid foundation for where they're going. Now, that's not the case necessarily for the big device companies, where they have much more resources and they can perform studies that span 5 or 6 years, which is typical for some of the bigger studies. The difference for us in, let's say, in startup versus big company is that most of the studies that tend to have the adaptive design, the devices have some type of an approval already, so they can get reimbursed for the devices in the clinical trial. Whereas for a totally new device, you cannot. It's very difficult to design a study that might last 5 or 6 years, which is how long these are taking. So there are some things that are afoot in the FDA over the last 2 years to try to help, especially smaller companies, to overcome these things, like the early access pathway, which is shifting some of the burden of proof from the pre-market to the post-market stage, and that poses new issues with trial designs of how do you really get real evidence in a post-market study? Very difficult. And then there's also the early access pathways, which are allowing first-in-man studies to be done in U.S. and discouraging people from going outside of the U.S. to do the first-in-man, like the phase 0 studies that you talked about, comparable in the device world.
Gwen, do you care to comment on that?
I think that there is more acceptance of doing adaptive designs, but we need greater modeling for both safety and for the endpoints of what we're expecting to see, because I think regulators appropriately are asking, how do you know that that's the right target population, and how good is the biology to tell you that you should be adapting and only treating this group of patients versus a broader group of patients. And that you might be missing important safety signals or important efficacy signals if you don't have the appropriate background biology to do the adaptation and the modeling of how you adapt.
Nicholas, would you care to comment?
Sure. So, I'm a believer in adaptive trials. I think one issue that, again, the community needs to be sensitive to or try to overcome is that most adaptive trials, by their design, leverage data that's generated within the trial to make decisions about which way to adapt. So if the basic design is something akin to a play the winner rule, then you, again, move patients or give them higher probabilities of being randomized to the drug that looks like it's actually benefiting more patients. But that's all based on the data that is being accrued within the trial. It ignores information that might come externally to the trial. So if there is, after a trial is initiated, a Nature cover article suggesting that one should use a certain drug in certain situations, and you're told you cannot take advantage of that information because you have rules in your trial locked in, then ethical issues arise. So I think, again, trials of the future are going to be adaptive, but leverage all sorts of information that needs to be, again, vetted at some level, but can't be ignored for ethical reasons.
Alice, would you like to comment at all in the imaging world?
I think everything was said. Probably one thing we need to be thinking of is that most of the trials now do not happen only in the US, they happen worldwide. I think, and we have a big disparity between different countries and different parts of the world. And by big disparity, I'm thinking in particular if you're thinking about China, or if you're thinking about India, which has a very large population, which is an untapped population for clinical trials. And that is something that we need to be thinking when you start big studies like that.
Yes, Dan.
One more comment, which I forgot to mention, was as opposed to a true adaptive type strategy, one thing that FDA has been encouraging is Bayesian approach, which is, and there, so far I'm not aware of a device that's been approved, but I know that there are several trials, including one that I'm involved with, where you have done a trial, the trial primary endpoint is negative, you identify a subgroup, and now you do another trial, and you borrow data from the original trial. The danger of that is most subgroup analyses, retrospective hypothesis-generating hypotheses don't bear out in the next trial. But anyway, the FDA is trying to be flexible on a lot of different areas because they know how hard this is. So the Bayesian, it's kind of a different approach.
Okay. So in the era of precision medicine, a call for the adoption of one-person trial designs, such as N of 1, has emerged. What are the advantages and what are the hurdles to adopting this type of approach? And I know, Nicholas, you have written extensively on this, and perhaps you can start the conversation.
And I'm an advocate of these trials, obviously. I've published such. I do think they have their role. I mean, we started out focusing on these trials in the rare disease situation where you simply didn't have the luxury of a large denominator to carry them out. And people have had success pursuing those trials. Well, there's no reason to believe that the kind of mentality that was brought to bear on the design of those trials in the rare disease settings couldn't be extended or generalized. Now, by N of 1 trials, I mean a real focus on individual patients, monitoring them with large amounts of data. But there's no reason why the results of multiple N of 1 trials can't be aggregated. I think one thing that I've been charged of is, or charged with in advocating these trials is not really paying attention to the need to generalize. But that's in fact not the case. Everyone could benefit from an N of 1 trial. And then you aggregate the results. You could identify more unequivocally who does and doesn't respond to a particular drug. So think of these trials as bringing out a phenotype that otherwise would be very hard to bring out. So once you have conducted a series of N of 1 trials, you may, again, identify responders and non-responders to particular drugs with greater precision. Then you could ask questions about what those patients have in common. But if you do not spend enough time bringing out the phenotype and monitoring the patients, you won't be in a position to say, unequivocally who did and didn't respond and therefore might share, say, a genomic profile or some biomarker.
Gwen, do you see pharma, or at least in your neck of the woods, adopting N of 1 or single patient trial designs and using meta-analytic techniques to build the power of your observations going forward in any particular studies?
I think that's going to be a hard sell for big pharma. And the reasons for that are multiple, not just financial. The toughest part is that there is an assumption that the drug that you're using, you already know the appropriate dose in that disease, and that that it doesn't need to be sequenced or given in combination or given multiple times or given over a prolonged period versus a short period. There are so many early signals that you have to answer before a drug is ready for that kind of a trial. And even then, it may be disease-specific or genomically specific or epigenomically specific about whether a person will respond to that, even if you've hypothesized correctly. And I think this is part of the problem with the I love basket trial designs. I think this is really very exciting, but it's so simplistic right now. We're looking at a single target for the most point and saying, okay, EGFR or P53 or whatever the target is, and almost every cancer, at least in my experience, has not been driven by a single target. We can't use the BCR-ABL model of tyrosine kinase inhibition for all cancer, and I think this is where we need to put some very clever thinking about how much can we do in silico to tell us about combinations and the right the right way to treat these so that the baskets are much more meaningful for the patients and not just answering a single pathway question.
So let's go to that. We heard in the last panel about de-risking targeted therapeutics, and we know that biosimulation has actually markedly reduced therapeutic candidate attrition, even though it hasn't eliminated that, due to poor absorption or distribution, metabolism, excretion, known as ADME. And the extension of biosimulation to the generation of virtual twins or avatars of patients through integrating multiscale biology and omics could create cohorts of virtual populations that could be manipulated in silico and perhaps eliminate some aspects of phase 1. So do you think that this is possible, likely, happening, going to happen in the distant future, near future? Your thoughts on this would be of interest. Who'd like to take the start? Daniel, you want to?
I can't comment on the pharma side at all, but in the era of heart failure and device-based therapies for very ill patients, I think it's going to come. That means, and specifically, we have the knowledge to simulate the hemodynamics and cardiopulmonary status of individual patients and then impose different types of therapies to understand prospectively what is the right therapy for a given patient. There are many devices that are used in critically ill patients that there are no guidelines, there are no clinical trials. The FDA approved, all the devices are mainly used off-label. They're approved for, like, for example, 6 hours where they're used for 5 or 6 weeks at a time. So we don't have the proper guidance. And one of the projects that we're working on is exactly this, is to simulate individual patients in silico and then use those simulations to guide therapy for knowing, and then, of course, matching that with our understanding of what the devices do. So I think that is a very realistic goal.
And Gwen, do you think that this will eliminate the need for traditional phase 1 approaches?
No, the element of surprise in phase 1 is never going to be eliminated.
Nicholas, would you like to comment?
Sure. So I also agree that this is in the offing. I think in the future these sorts of activities will occur, and I say that for 2 reasons. And one just has to do with the fact we live in this big data era. So lots of data will be collected at individual patients that can be leveraged when combined to design simulation studies and whatnot. And even in the context of these N-of-1 trials, I'll just mention that I've been interacting with people with the FDA about these sorts of designs. Whether they're ready for prime time in pharma or in other areas is an open question. But one person in particular, Sean Cozen, who's there, who really focuses on oncology, has made a distinction between kind of narrow data trials and wide data trials. And narrow data is a few data points on thousands of people, and the wide data trials are thousands of data points on a small number of people. And with enough data points on individuals, you might be able to model physiology better, combine those data from different patients again, and then draw inferences about more generalities and whatnot, and then use that as maybe fuel for some of the simulation-based studies that are likely to emerge.
Are any of these in silico prediction models used for neurodegenerative and imaging approaches?
We do that for imaging. I think we not so much to make sure that the compound is. One of the main reasons is to eliminate the compound that will not work. And I think we are very lucky in imaging that we, at least for CNS imaging, that we can work with non-human primate, and that is a very good model. Doesn't work all the time, but I would say 99% of the time it's a good model. So clearly moving from in silico to eliminate most of the compound that will not work, and that is happening right now, to move into a very small number of animals, then to even smaller number of people, because we need to go into human to show that it's working. But yes, it's something that we do.
Another question that comes to mind is there's been increasing interest in that's evolving in industry, regulatory bodies such as the FDA, patients and stakeholders in leveraging, excuse me, real-world data such as electronic health records, pragmatic clinical trials, and other types of information generated outside of a traditional controlled clinical trial setting to speed the development of new therapeutics and expand indications. What do you see as the critical hurdles standing in the way of greater development and reliance on real-world data? And what can academic institutions and industry, either alone or in partnership, do to advance that cause?
Well, I think this is very relevant for devices because, as I mentioned, there's this new pathway, the early access pathway at the FDA, which is trying to get devices out into the public quicker, which shifts the burden of proof of feasibility and safety from the pre-market to the post-market. So there need to, and with that, with those such approvals, with really limited information, you may get a device approved with as little as 300 or 400 patient experience. There's a partnership with the FDA that you are mandated to do a much more robust post-market surveillance. So the question then is, okay, I can measure my outcomes in a patient population in real world, but then what do I compare it to? So this is a real problem, and this is, not, the devices are just starting to go through this pathway, so we don't really know the full answers. For the most part, these rely heavily on the development of registries. And so that's one part, is to establish the infrastructure to collect the appropriate information from every patient that gets a certain device. So there are registries, for example, at the ACC and the surgical societies. There are some registries for certain devices. The rigor with which the data are collected is very variable. The data are also usually not monitored. So that's a big issue, and of course it gets back to funding. Who funds it? Usually the hospitals have to pay into the registries to have the privilege of implanting these devices. So there are big issues that need to be addressed. And then the clinical trial design, the data analysis plans are really not well established. Do we do the propensity matching with other cohorts? Do we do just objective performance criteria, which say, okay, you have to meet this bar? But this is totally unexplored and really needs to be worked out.
Right. Gwen, I know you have some thoughts on this.
So I think there are two important points, and I think one of them is that we all know that looking at the medical literature and trying to come up with what is, if you're doing a single-arm trial, so you're trying to use the least number of patients so that you don't expose more patients than absolutely necessary in the trial, especially in an oncology setting where this may be their only chance and they may be dead before their next chance comes. You want to minimize that human risk and human cost. You look in the literature and you say, okay, in relapsed refractory DLBCL, what is the appropriate response rate I should be looking for with my agent? And you find that it ranges from 20% to 60% response rate. So you need to either have an internal database that you can compare patient to patient to, or you can look at a real-world virtual control arm, which is what a lot of pharma companies are hoping to be able to do with increasing use of real-world data. That brings me to the second point, because I think that's excellent, and I think that there's real reason to do that. But the second point is, we talked a lot about the quality of our targets and how we look at the target quality. We also need to look at the quality of the kind of data that's being collected. And most of the real-world data sources that I've had the opportunity to look at are spotty at best, and are really we're not taking advantage of the great electronic, ways that we could be collecting data from patients. There are huge ways that we could improve this collection and train the people who are collecting the data, which is another thing. We've neglected data management and nursing and some of the key players who do this for us in how important they are to understanding what we have in these databases.
We're going to We're going to go to some of the audience questions that we've received electronically, and if those in the audience have questions and want to come to the microphone, that would be great. One question that was posed is, can you speak to how advances in technology, social media, quantified self, such as and phone apps have influenced the collection of data in clinical trials or are being utilized in the development of endpoints for clinical studies?
Sure. I think they're going to have and continue to have a large impact on the way trials have been done. I'll just mention one trial I was involved in recently having to do with a local company called Click Therapeutics, which is right in Manhattan. The trial was done 100% in a remote fashion. Now, it used a series of apps in a smoking cessation application or setting, but everything was done remotely. The data was collected, the patients were consented, et cetera, et cetera. Again, you can debate the merits of whether the trial design resulted in valid outcomes, but suffice it to say there was an attempt to put together a trial that was completely remote using nothing but applications on a wireless device.
I saw a great neuroscience app demonstrated at a Roche Science Days for Parkinson's, which just uses your iPhone, right? And you know that every time that someone comes to the hospital to see their physician, they either are much sicker than they were at home or they're much better than they were at home. They're never actually at that continuous level of wherever they are with adverse events. And we're trying to collect this data. Well, you hold the phone and you see how much your hand is shaking, and it can do the oscillations. And you do this at set, it bings, okay, time to do your hand, and you can check the level of Parkinson's symptoms. These kinds of clever ways to actually get real patient data in their home setting is so much. We're just touching the surface.
We have a comparable thing in heart failure where a company was, if you have heart failure, you call up the company phone number and you speak a sentence, and it analyzes your voice in terms of whether you're fluid overloaded or not. And so they're exploring this for COPD and heart failure, so it's another cute little thing.
Bob?
Janice, I might have missed this because I stepped out at the beginning, but we talked a little bit about last night at dinner about how do we get beyond standard of care, and the regulatory burden for that. So, for example, in diabetes, sulfonylureas and metformin are in the water, but that leads to a really bad outcome at the end. You're going to get refractory disease and nephropathy, things like that. And you need $1 billion clinical trial to prove that you're better than this bad outcome. Or, for example, in oncology, standard of care, your first entry is into a refractory patient who's really, really sick. So how can you leapfrog that? Is there any way to think about leapfrogging to get a truly novel therapy out there?
So how do you really match the appropriate target population to your newer approach early on in the development to have a better impact? Any thoughts?
Again, I am kind of representing my own views, not the views of my colleagues. But I think this actually happened spontaneously, but could be helped along. And I'm not an MD, but I was going to say, but I play one on TV. That's actually not true. But I work with many. And, physicians are given license to practice medicine, and they can draw on different things to make conclusions about how best to treat their patients. So, for example, I was working with someone the other day who came home. We had a dinner meeting and struggled thinking about how best to treat this patient and were noticeably confused about what to do. So they did what I think most physicians or researchers would do. They consulted with colleagues. They went to PubMed. They did a Google search. They didn't have time to read every article, so PageRank gave them 5 articles that they might be able to read that were ranked top. On that basis, they kind of came to some conclusions, and the next day moved forward. Nobody's formalized those sorts of things, and yet they happen spontaneously. So the degree to which PageRank got this person the articles that would benefit the patients most is an open question. But you could imagine trying to formalize this, put forward in front of the physicians things that might benefit them. What those might be is debatable, but this is the way to actually influence the clinical decision-making. And whether one could conduct clinical trials to see the virtues of PageRank versus some other way of ranking algorithms, again, is an open question, but it will happen, and it is happening.
I think the process of identifying safety, we talked a little bit about this, the use of avatars or virtual populations to really do that testing in silico would allow one perhaps to more ethically enter into a more wellness population earlier in development of a reagent. So we did spend some time talking about that as one way of bypassing that template.
Thanks.
I'd like to, one other question that has come up is what are private academic partnerships that emulate best practices for allowing for acceleration of discovery science all the way to approval that you are aware of that we should try to model or that are informative in terms of how we go about this process?
I think for Parkinson, that one that I'm very familiar with, which is the PPMI, which is funded by the Michael J. Fox Foundation, which is a public-private group of studies that happened, and the studies are run in Australia, US, and Europe, collecting many samples, recruiting something like 1,500 subjects that are going to develop Parkinson. I think that is one. The other one, as a consortia, it's something related to what Michelle said, which is ADNI in Alzheimer's disease. I think that are two kind of consortia that are very powerful and that help the industry and the academic world at the same time. And I think probably, so that are the two I'm aware of from the neuroscience point of view.
Great.
But there's broader infrastructure, and it hasn't been mentioned, the Clinical Translation Science Award program from NCATS. This is a $750 million effort to build translational centers within academia whose goal is to promote translational research, where one goal is to encourage industry-academic interactions. Again, I've been involved in that for about 10 years. Its successes can be debated, but it is an overt attempt to engage and build a network to reach out and interact with industry.
Another one to mention might be the recently announced Beat AML study with the Leukemia Lymphoma Society, where having a foundation be the sponsor allows multiple pharma companies with multiple different lawyers to actually participate in a basket trial for a disease where the academicians are part of picking the targets and then participating in the trials, and the companies have a role in getting back science from the academic centers. It's a very nice way to treat a disease where at least big pharma has a lot of hesitation.
I think Cystic Fibrosis Foundation is another one, is a good model, and I think that the NCATS CTSA approaches are really building on these really outstanding models that you all commented upon. I'd like to thank really our outstanding panel members and the audience for their participation, and we'll be moving on now.